Hello, everyone. I'm Paolo. I'm from Denmark, working at DTU, Technical University of Denmark, on antibody and T-cell receptor structure mainly. And today I will be presenting two tools that are LYRA and SCEptRe that are connected to retrieving and modeling antibody and T-cell receptor structures. You can get access to these tools by the IEDB page through those links, T-cell tools and B-cell tools. Here they are. And I will start talking about SCEptRe. The main idea is that we are starting to have quite a large number of T-cell and B-cell receptor structures available. And the first tool that I will be presenting, that it's SCEptRe, it's a tool that is used to retrieve and filter these experimental structures. Why we need to do that? So first of all, very often these kind of resources, the main database that we use to get access to short structures is the PDB, is the protein data bank, are updated with a very high frequency. So we want to get databases that are, or data sets that are updated in an easy way. But also we want to get some control over the structures that we retrieve. So we want to have a given quality of the structure. We want to be able to get structures of different antibodies and T-cell receptors, and that's why we need tools to automatically retrieve those structures. And the way we filter structures is based on the antibody and T-cell receptor specifics, meaning that the way we want to be able to filter them is not based on the overall sequence, but it's based on specific regions. You all know, I mean, the overall antibody and T-cell receptor structure. We have here a picture describing the structure itself. Here we have a T-cell receptor in gray. In light gray, we have the alpha chain, and in dark gray, we have the beta chain interacting with a peptide MHC complex. This is a class 1 MHC, in which we have an alpha chain and a beta-2 microglobulin presenting a peptide here in the middle in orange. And you see that you have very specific regions of the T-cell receptor that interact with the MHC and with the peptide. If we zoom in on the T-cell receptor, and I'm looking here at the right, we can see that the interaction is mainly driven by these 6 loops that are colored in shades of blue and red, that are the so-called CDR loops, complementary determining regions. We have 3 on the alpha and 3 on the beta, and similarly, we have 3 CDR on the light chain and 3 CDR on the heavy chain for antibodies. These 6 loops are the ones that give the specificity. So you have a pretty much conserved structure all over the place, but these 6 loops are extremely variable in sequence and to some extent also in structure. So whenever we want to create a set of structures, we really want to focus on CDR loops because those are the ones that really are important for the function. Why we need some tool to filter things? Well, why I need this kind of tools, I can tell you why I am always using this kind of tools, is that we want to develop statistical models or machine learning models for predicting the behavior of antibodies and T-cell receptors. So we want to have a way to understand the specificity by looking at available data. And one of the problems is that, first of all, you need to be able to filter out experimental data of low quality. So you need to have a way to only retrieve data of good quality. But also, and this is a major problem when you develop computational tools, you want to be able to control redundancy. If you work with antibodies and T-cell receptors, you know that there is a large number of structures for a very little number of cases. So, for example, COVID or HIV, we have plenty of antibodies and T-cell receptor for these cases, but maybe we have very few for other epitopes. So we want to be able to control that. Redundancy is in general not a good thing in data sets, and especially when you want to develop models, redundancy has a major impact. It can cause one thing that is called overfitting, meaning that our model will not be able to predict nicely on all cases. And in general, if you want to understand or to compare your own sequences of antibodies and T-cell receptor, and to see if there are structures available, you want to be able to have a unbiased overview of similar sequences. This is why we need this kind of tool. Just to give you an example from the IEDB, if we look at CDR3 sequences of beta chain of TCR that are targeting this peptide GLC, TL, VAML, we can see that there are plenty of TCRs that are very similar, meaning that their CDR3 is almost identical with 1 or 2 mutations. And this is true also the other way around. So we can have cases in which we have that a single TCR binds to peptides that are quite similar. So we want to be able to take these things into consideration. That's why we need a tool like SCEptRe. This is the main interface of SCEptRe. We'll start with an interface with the first page in order to retrieve TCR structures. And as you can see, this is you have a number of fields that you can use to filter. And the main idea is that, as I was mentioning, you have two main goals: to keep a given level of quality for your structures, and to be sure that you don't have a bias in your datasets to keep a given level of redundancy. The first part, that is the first 3 fields in the upper side of the form, are connected to the experimental quality. Most of the structure are X-ray structures, so we have that the main parameters that we can use to filter the quality are the resolution, the R3, and the missing residues. If we think at a crystallographic structure as an analogy, as if it were a picture, the resolution is actually the resolution of the picture, the amount of information that we have. So the lower the number here, so 1, for example, it means 1 angstrom, that it's a measure of the resolution, the larger the amount of data that we have. So it's a finer, yes, picture of the structure. On the other hand, the R3 is a measure of the noise that is present. So how well the structure that we have in the PTP is actually fits with the experimental data. And in this case, again, the lower the number, the better the quality. Finally, we have the fact that we want or not to include TCR or in general molecules with missing residues in the CDR. And this is again connected to the experiment. In some cases, there are residues that are invisible in crystallographies just because, for example, they are flexible and they move too much. So they are out of focus in the picture, let's say. And in general, we don't want to include those to be on the safe side, but there might be cases in which you actually want to have a larger data set and also include those. So again, resolution, the lower the better, and the default is treat that it's a kind of a good compromise between amount of structures that you can retrieve and quality. They are free, the lower the better, and the missing residues. The second part is about the type of molecule that you want to retrieve. And you have the first drop-down menu that allows you to select between peptidic epitopes and non-peptidic. The vast majority, of course, are peptides, the type of epitopes that most of the classical MHC bind. But of course, there are also a few structures of non-peptidic epitopes. If you select peptidic, then you have the option to filter on the peptide itself. You can filter on the length of the epitope. You can filter on the redundancy, so you can kind of cluster together structures that bind, in which the TCR binds to very similar peptides, and you can cluster on the binding core of the epitope or on the extended region of the epitope. This is mostly important for class 2, in which you have that the binding part, the region in which the epitope binds to the MHC, it's just a smaller region of the complete peptide. And the sequence identity is really important. Why is that? Because we know that, for example, when we develop models for TCR specificity, those models are usually quite good at predicting specificity to peptides that are very similar or identical to the peptides that we have in dataset. And they are less good when we move farther from that. So it's a good way to understand the data that we're looking at and how well we can have rules or analysis that generalize to many different peptides. Finally, you can also filter on the organism, that it's the source organism and the type of MHC, class 1 or class 2. Then you press submit, and you have an output page that is like this, in which you have plenty of information. And this is a kind of type of table that you have seen several times. The specificity here is that since you can cluster by identity, the first column that it's named cluster is giving you an identifier of a number of structures that fit with the criteria that you have selected. So in these cases, you have a cluster ID that it starts with a first number that is connected to the epitope. Then you have two numbers that are connected to the alpha and beta chain. And then you have finally some numbers that are, or numbers and letters that are connected to the MHC. And as you can see, for example, in this case, you have 4, and 4 is connected to the epitope that is GIL. Then you have A1, B1 connected to the alpha and beta chain of the TCR. And then you have A1 that is connected to the MHC. If you filter the epitope at lower sequence identity, several different peptides will fall into a single cluster. Then you have other columns that link to IEDB pages with more information about the specific sample. They link to the epitope ID and to the receptor ID. You have also links, internal and external links, to actually visualize the structure, either by going to this view 3D structure that will open a new tab or a new page to visualize actually the complex, the TCR and the MHC with the peptide. You can also play around and visualize interaction of specific region, or you can open the external link to the PDB with the original data. And yes, you can download the results with the link that it's up there with download results. And basically the same is true for other pages. So for example, if you want to retrieve MHC complexes bound to peptides, you have more or less the same interface with resolution and are free. Again, you can select peptidic or non-peptidic. Again, you can filter on the length and filter on the organism and MHC class. And the output page is more or less the same. In this case, we don't have information about the TCR because you might have structures in which the peptide is bound to the MHC, but there's no TCR in the complex. Again, the same is true for antibodies in the BCR, B-cell receptor tab. You can filter on the same parameters. In this case, there's a small difference in which, again, peptidic and non-peptidic in this case means that it's either, if it's peptidic, is it, the antigen is either a peptide or protein. If it's non-peptidic, it might be a small molecule or some other kind of molecule that it's not a protein. And you can filter on the antigen length. And in this case, if it's less than 50, it's typically a peptide. If it's 50, 60 or more, it's a protein. And more or less the same things that I've described before apply to this output page. Now, of course, this is a very useful resource, but the amount of structures that we have in the PDP, the amount of experimentally solved structure is pretty minute if we think about the great diversity that we have in the T-cell receptor and B-cell receptor repertoire. So what if the molecules that we want to analyze are not in the PDP? And just to give you an idea, we have at the current time approximately 2,500 antibody structures and probably a few hundred, 300 or 400 T-cell receptor structures. If you think about the diversity you can generate in a repertoire, this is a very minute number of structures. Does this mean that if there's no structure available, I mean, we cannot include structure analysis in our work? This is not the case because luckily we have very accurate tools to predict the structure of T-cell receptor and B-cell receptor. And this is the other tool that I'm going to present, that is LYRA. LYRA, that is the acronym for lymphocyte receptor automated modeling, is a very simple tool to model the structure of antibodies and T-cell receptor to a very high accuracy. And the main concept here is that when you do standard, let's say old school modeling of structures, I'm saying old school because we all know that now AlphaFold has kind of revolutionized the field of structural modeling. But in general, when you model proteins, your model is, the quality of your model is constrained by the quality and the availability of similar proteins that have been experimentally solved. So in general, if you have regions in your protein that are very similar to something that has been solved, then your model will be good. And if there's no similar structure, no similar sequence, then the model will be less accurate. But in the case of T-cell receptor antibodies, the part that is less similar, that is the CDR loops, is actually the part that is responsible for the specificity. So you can have a very good model for the conserved framework regions. And then you would have a less reliable model in the CDR loops. And this is really undesirable because your model would be almost unuseful. But luckily in the 80s, Cyrus Chodia and Arthur Lesk discovered that the sequence variability of antibodies and T-cell receptor in the CDR region does not directly mean that they have a huge structural variability. And they created this theory or this protocol. This is called the canonical structure protocol. That means that you can actually predict the structure of the CDRs to a very high accuracy in the vast majority of cases. And this important discovery has generated a way to create antibody anti-cell receptor models that it's highly accurate. The main idea is that you can actually exploit these limited variability of the CDR to create accurate models for the complete structure. This is the main page for LYRA, in which you have two text areas in which you will just copy and paste the sequence of the LYRA and heavy chain or the sequence of the alpha and beta chain. It works both for antibodies and T-cell receptor. It doesn't matter the order in which you put the two. It's important that you put amino acid sequences or with or without the header, so either in FASTA format or in pure text format. And then the tool, if you don't want to go into details, can work without any additional parameter. And the main idea is that the framework regions, that is everything that is not the CDR region, is modeled by homology. So by identifying similar structures in the PDB. For the CDR, the tool will first identify what is the canonical structure, so the overall shape of the CDR, and then identify a template within that specific class. So a template that is known to belong to that specific, to have that specific shape for that specific loop. So yes, you just upload the sequences, as you see. It can either be in FASTA or plain text. In case you need, there is an advanced option tab, and you can select basically a few more advanced parameters, mainly related to the modeling of side chain. As you know, the proteins are, in general, amino acids are composed by a backbone or main chain and a side chain. And in general, the modeling is accurate for the backbone and is less accurate for the side chain. So in specific cases, you might want to play around with these. And the main idea is that here you can use the default method that is used by LYRA is to copy the side chain of residues that are identical in the template and in our target protein, and to model the ones that are different using a tool that is called SCEptRe, that it's a sidechain modeling tool developed by Roland Dambrecht. You can also decide to exclude specific PDB structures, so experimental structure, from the modeling protocol. And this is usually, let's say, this is something that you can use either if you want to benchmark your tool. So you predict a new structure that has just recently been solved, and you want to see how good you can perform. You blacklist that specific structure so that it's not included in the modeling, and then you can basically assess the accuracy of the modeling protocol. Or if you know that a specific structure has some feature that is undesirable, for example, it has mutations that have been introduced, or it has a quality that is not satisfactory, you can actually blacklist that just by writing the PDB code for that specific structure. You submit, and the output page looks like this. You have several tabs. In the first tab, its result. You just get your input data. Then you can get an idea of the alignment here. These two are the alignment of the light chain and heavy chain, your input sequences, and the template sequences that are being used to model that. And in the upper table, you see the actual templates that have been used to model the framework of the two chains and each individual CDR loop. And as you can see, you can use multiple templates, up to 9, to produce a single model. Then you can visualize your structure. As before, you can color different regions in a different way. And to give you some like rough estimate of the accuracy, in this plot, we show the accuracy for B-cell receptors and T-cell receptor, antibodies and T-cell receptors. For antibodies, each column is representing a different region, L1, L2, L3, all the loops and the framework. And here is the average accuracy, the average error-density, meaning how far away is the predicted model to the real structure. This is done in a blind assessment in which the specific structure has been removed and then predicted using all the rest of the data. And as a general assessment, you can see that you get an accuracy of the overall framework that is less than 1 angstrom. And for each loop, in general, that it's below or approximately 1 angstrom. And the green bar shows the ratio or the fraction of models that are of extremely good quality. And then up to red, in which you have red and black that are poor and very poor quality. You see that basically around 80–90% of the structure are of very good quality, and the green region is showing qualities that are almost as good as the experimental quality. This is also true for TCR, less than antibodies. So the quality of T-cell receptor models is slightly lower than antibodies, just because the number of templates, the number of salt structures for T-cell receptors is lower. And the exception is the CDR3 of the alpha and beta and LYRA and DEBI. This is still, the quality of these two loops is still lower because they are more variable. Also their structure is more variable. So we have a slightly lower quality for these regions. But again, we envision that with the new tools that are available to model structure, like AlphaFold and other tools connected to these deep learning methods, the numbers, the accuracy of the H3 or CDR3 in general will increase in the next few years. Why is this important? And I'll quickly give you, let's say, an overview of what you can use this for. We have also developed a DTU tools to predict the structure of complete T-cell receptor peptide MHC complexes, and we have shown that one can actually use these models to improve the prediction of specificity for T-cell receptor. So if you model the structure of a complex TCR-PMHC complex, and you put it together using, for example, the TCR-PMHC tool that we have developed a few years back, and then you use computational force field to measure the interactions between the T-cell receptor and the peptide MHC complex, these predicted energy that you get from the force field can actually improve, help you to identify what is the right epitope that a given TCR binds. We have used this force field, and the force field that we use are the Rosetta and Fold X force field, to try and pair together a given TCR with the corresponding peptide, and we see that we can actually use the force field and tweak it and fine-tune it to be able to recognize to some extent the interaction between TCR and PMHC. It's important to underline that this prediction, of course, is based on a structural model and a computational force field, so it's not very precise. But the good thing is that if we use this as an extra information to identify, for example, which TCR binds to a given peptide, so we want to pair a given TCR to a very specific peptide MHC complex. The most naive thing that we can do is just to use the MHC binding affinity and to say, okay, we just predict the specificity based on the MHC peptide binding alone. And in general, we get good results because the MHC is actually the the MHC binding is the most discriminative part of everything. But if you, if you also include the information about the binding energy between the TCR and the PMHC, you can actually slightly improve the prediction. So here on the left, we are only using information about the TCR binding from the models that we generated. On the right, we're only using MHC binding. If we mix the two, we can improve the performance. And this has been kind of included in different pipelines. For example, in pipelines for rational epitope discovery, or to design antibodies, therapeutic antibodies and T-cell receptor. And the main idea is that you can use these to generate models, to analyze the interaction, and then to propose mutations to the antibody or to the T-cell receptor to increase the affinity, to perform experiments, and then in a iterative way to improve your molecules. So to sum it up, I've presented today these two tools, SCEptRe, that gives you access to PDB structures, to experimental isolate structures of B-cell receptor and T-cell receptor, and allows you to create customized data set with a given quality and given redundancy. And then LYRA, that it's a tool that you can use to model T-cell receptor and B-cell receptor without high accuracy. And this can be used to perform a large number of downstream analysis. And that being said, yeah, I finished with my presentation, and I leave the word to Nina.